This is podcast number 42. So, we've been talking about how cells get energy and convert energy from one form into another. We talked mostly about how it converted glucose into ATP, and then we talked about how plants take sunlight and make their own ATP. But there are other organisms out there that don't do either of those things, and they tend to live in places we don't associate with supporting life. Those are called extremophiles. They love, or phile is the part of the word, they love extreme environments. There are bacteria that live in acid mine drainage that is as low pH as like inside your stomach. There are bacteria that live on uranium. They're shown here. That color green is false color. It's not like they're Hulk bacteria or anything like that. It just they color them in the image so that you can see them better. There are bacteria that live at the bottom of the ocean and support bigger organisms, and there are bacteria that live in toxic waste dumps that give some of the colors that you see here. If you've ever been to Yellowstone and you see some of those vent pools where it's really hot, are really acidic, are really salty, there are bacteria that live there too, and they give those a color. How acidic is it? You know, every year there's stories about people who don't pay attention to the warning signs, and they walk off the warning signs, and then they fall in those pools, and they die. They don't recover the bodies because there's no body to recover. They just dissolve. So these are really extreme environments that these bacteria are living in. Okay, so there are bacteria that eat methane, there are bacteria that eat rock, bacteria eating toxic waste. There are even bacteria that are under the bottom of the ocean. So looking at DNA at different depths, all the way 1.2 miles underwater, and then going underneath into the sediment underneath all that water. Even 6.6 meters, 20 feet or so into the sediment, still detecting bacteria as detected by DNA. Another study looked at actual cells, took samples and looked at cells under the microscope and counted them, and plotted them according to plotted them according to a log scale. So this is 3.24 miles underwater. And then under that 3 miles of water, then go even further into the soil. This is not as far as the previous study because this is in centimeters. But even so, you can see that there are lots of cells underneath all that water. In cold seeps, which is a special type of environment under the ocean that seems to be associated with a lot of life, there are even more bacteria per unit of sediment. Do they all eat the same food? So the photosynthetic plants that we talked about are called autotrophs, and they make their own food. Heterotrophs eat something that somebody else made, like we are heterotrophs. And then there are things that take chemicals from their environment and they use those as fuel, like we take chemicals that are put together by plants and we use those as fuel. That makes us heterotrophs. But there are other ways of making a living other than plants and animals. and if you plot how many microbes there are in a log scale here on the red line. So right here would be 1,000. They don't tell you per how much sample. So they typically grab a core out of the sediment, a circular coring drilling machine, and then they analyze a certain amount of that, but they don't tell you how much. But in whatever sample that is, they detected 1,000 bacteria, for example, right here. And then as the line dips this way, it's more, this way, less. Right there, that line is 10,000 cells. And it's not that, when you get really deep, the number of cells drop off. They don't. So the number of cells that you get right underneath the surface, just 2 meters underneath the surface versus 80 meters under the surface, not that different. And if you look at what they eat as you go from the surface down, it tends to be that you get more and more heterotrophs. This white bar becomes more and more prevalent as you go deeper. But this is just one study. In these cold seeps or hot thermal vents, you have a part of the ocean floor that is in contact with the mantle, not very far underneath it. So lava that superheats the water, and superheated water can dissolve lots of minerals. When that superheated water hits the cold water at depth, let's say this is a mile below the surface, that water is going to be cold. Then those minerals fall out of solution, and they sort of look like smoke. So it's not smoke in that you would see smoke from burning something. It is just minerals that precipitate out of solution as the superheated water hits the hot, superheated water hits the cold water of the ocean. So the superheated water is in the bedrock, and then it hits the cold water when it comes out, and you get this sort of vent sort of geography. And around these, you have dramatic life. And I remember being in school when these things were discovered, and it was pretty unusual. It was sort of like discovering aliens or something like that, because these sorts of things hadn't been seen before, these giant tube worms. And then there are crabs and shrimp and fish that live near these systems. And to give you an idea, like, what do I mean by superheated? Well, water boils at room temperature and at 1 atmosphere of pressure at 100 degrees. This isn't at 1 atmosphere of pressure. This is several atmospheres of pressure because it's underneath a whole bunch of water. But it's like 300 or 400 degrees centigrade, that water that's coming out of there. And then it falls off quickly. So right where that water is coming out, it could be 300 or 400 degrees centigrade. And then a foot away from it, it could be 10 degrees. And then 2 feet away from it, it would be below zero, actually. You can be below zero and not form ice if you're under great pressure. So it's a big steep gradient of temperature. These subs that get these pictures, these research subs that get these pictures, have a giant plexiglass dome so that they can see it. And I've heard pilots of these subs speak before, and they said they have to be really careful to not get too far or too close to these smokers because you can melt the plexiglass. So they're careful about that. if you look at a shrimp, like a shrimp from a shrimp cocktail that you might eat, it has an eye, antenna, and so on. If you look at shrimp from these thermal vents, they're white. They first thought that they were blind because they don't have traditionally what you would see as an eye, but they do have this area where an eye would be, and it is innervated. It has neurons going to it, and so the question was, is this a leftover of evolution, like they used to have eyes, and they are just being selected against, and we are seeing the remnants of eyes, or do these eyes actually work? And if they work, what are they going to see? Because there shouldn't be any light that deep in the ocean. So they extracted pigments from these things that might be eyes, and they got a pigment that looks just like the pigment that is in a vertebrate eye. So this is rhodopsin from a frog, and the pigment that they got in a shrimp has a very similar absorption characteristics. So there's like, okay, well, why do they have that if their eyes don't work? So then they started looking at pictures of what these vents look like. So if you go down there in your sub and you take a picture with a flash, you get this sort of thing. But then if you shut off all the lights and don't use a flash and just use some really sensitive CCD cameras, you still get an image. And there is still something like light that is exposing that sensitive surface on the CCD camera. And so what is that? It is light of some form. Then if you take that light into different parts of wavelengths and say, what kind of wavelength light is that? It is on the high end of what we see as visible light, and then some past that, called far infrared. Okay, so right on the edge of visible light here, okay, and then past that here. And infrared is what we associate with heat. So if you go out in the sunlight and you feel the warmth of the sun on your skin, you're detecting infrared light. You can't detect it with your eyes, but you can feel it as heat. And maybe that's what those shrimp are using to see. Don't exactly know. But there also could be some light that is not being picked up by these instruments. So if you calculate how much light there is according to these sorts of pictures and say, okay, we're getting this much at this wavelength and this much at that wavelength, you get that sort of a curve. This much light of this wavelength and this much light in the far infrared. But if you actually observe how much is there using a very sensitive light meter that measures visible and invisible light into the far red, visible or invisible according to what humans can see, you get more light than what is calculated. So there's probably light there that's not being detected. Whether or not the shrimp can use that light, don't know. It certainly seems like they can. It's not surprising that another animal could use light that we can't use. Bees can see in the ultraviolet light, in the ultraviolet range, and you cannot see that. So when a bee looks at a flower, they see a different thing than when you look at a flower. You may look at a flower that is just white to your eyes, but to a bee it might have bright yellow stripes that lead it to where the nectar is in the middle of that flower. and hummingbirds can also see colors that we can't see. So it's not surprising that the shrimp that lives in this environment that we originally thought of as not having any light at all could actually be using what light is there to form images, or at least detect light. So these vent animals, like the shrimp here that we've been talking about, there's vent shrimp, there's tube worms, there are fish, all this ecosystem that lives at this around these vents is based on bacteria that live around the vents, and those bacteria are using not photosynthesis, we thought. Most of the bacteria are not using photosynthesis, but instead using the reduction of sulfur compounds to get their energy. So different than how energy is produced in photosynthesizers, different than how it's produced in animals. But then there are recently discovered bacteria that might be able to photosynthesize. So if you think about this, this is at the bottom of the ocean. We thought there was no light there. Maybe there is some light there. Take bacteria that collected around these vents, and you extract their pigments, and their pigments look just like photosynthetic pigments. So I showed you this a couple podcasts ago, where you say what at a certain wavelength, how much light do you absorb, and then you change the wavelength and you make this sort of spectral graph. And remember, green was reflected and so on, and not absorbed. So these pigments look just like a spectral graph that you get from a plant. And why would you have this sort of pigment in bacteria that live at the bottom of the ocean where there is not much light, even if there is light? So there is evidence that these guys are photosynthesizing. So they're using the light that is available, like you saw in that picture of the vent, and that is enough light for them to actually fix carbon. They don't get their carbon from CO2. They get their carbon from the water, but they are using the captured light to excite electrons and do an electron transport chain like we talked about in photosynthesis. So they may be using a different electron donor. Okay. So they how they get their electrons excited and what they're exciting might not be the same as an accessory pigment, or it may be. And they might be using a different electron acceptor. So the electron acceptor at the end of the photosynthetic electron transport chain is NADP. You don't see that on this list. They may be using a different electron acceptor, and they're doing chemosynthesis instead of strictly photosynthesis because they're starting off their electron transport chain from a chemical, not necessarily from light. Or in this case, maybe it is from light. But the point is that there are different ways that different organisms can use some of the same ideas, and that you're going to pass electrons down a chain. They're going to create a pH gradient, and then you're going to use that pH gradient to make some ATP. How that chain starts, how that chain stops may be different, and how you get your electrons excited may be different, depending on what kind of organism you are. But there are many different types of microbes that can do this in vents. Some scientists think that all life on the planet started at vents and then invaded the other parts of the ocean and land after that. But one of the ideas is that life started at vents. Another sort of environment where you see extremophiles is in very, very salty environments. Those are called halophilic. They like salt. Halo is salt. Okay, this doesn't mean you like the video game Halo. It means that you really like salt. And in the Dead Sea, there are no eukaryotes living, but there are prokaryotes living. They give the Dead Sea a pink sort of color. You can see that in some of the pools in Yellowstone also. And the salt concentration in the Dead Sea is 10 times what it is in salt water. So it's very, very salty. Because there is a lot of salt that is outside the cell to balance what's inside the cell, it's thought that the proteins have more acidic residues than is typical. So if you look at the amino acids that are in E. coli, and if you look at those same proteins and what kind of amino acids they have, they tend to be higher percentage of acidic amino acids in halophilic bacteria that live in the Dead Sea versus these guys that live, say, in your mouth. There are other organisms that we're just sort of discovering. We're discovering organisms all the time. This is an organism that lives in the open ocean. It doesn't even have a scientific name. It just has a number. And it has had its genome sequenced recently. It is photosynthetic. It is a cyanobacterium. It's a photosynthetic cyanobacterium. An example of cyanobacteria are like the things that stain your roof on your roof tiles, stain them black. Some of the algae that cause a bloom in Lake Erie and can make the water toxic to drink are cyanobacteria, but this guy is not one of those. So when they sequenced this genome and looked at all the genes, they calculated all the things that this thing can do in metabolism. And some of the things you recognize here, like here's glycolysis, right? And they can make fatty acids, and they can make amino acids and all that sort of thing. But some things are missing. Like there's no TCA cycle, no stage 3. And that's pretty unusual among organisms not to have a TCA cycle. There's also no Calvin cycle. This is a photosynthetic organism. It doesn't have a Calvin cycle, and it fixes carbon in a different way than the Calvin cycle that we talked about. The Calvin cycle is the one that takes CO2 from the atmosphere and fixes that in a 3-carbon molecule that the plant can put together to make 6-carbon molecules. It's not in this organism. So the point of this thing is there's lots and lots of ways for life to exist, and it could be that we will, and it's likely we will discover things that's going to violate things that we consider to be absolute rules. Its photosystem also has no photosystem 2, only photosystem 1. That's not so unusual. There are other bacteria that have only photosystem 1, but no TCA cycle and no Calvin cycle. That's pretty unusual. Did it have these genes and lose them? That's the hypothesis. it probably lost these genes because it could get away with not having them. They weren't a selective advantage. They were neutral for this particular organism in this particular environment. It's not parasitic, so it's not a situation where it's living in something that it's supplying a Calvin cycle or a TCA cycle for it. It's living by itself. So there was no selective pressure to maintain those genes if it once had those genes, and so they're lost either through drift or through mutation, and the organism does just fine without them. So the point of this podcast is that there's as we explore our environments more, we're finding organisms that seem to go about gathering energy and making a living, so to speak, in ways that we did not think were possible, like organisms that can see where we thought there was no light, organisms that thrive at really high pressures and really extreme environments, organisms that can photosynthesize without a Calvin cycle, without photosystem 2. And then this one, I didn't show you this picture. This is a bacterium that looks like it's splitting in 3 instead of splitting in 2, which is all the other bacteria we know about split in 2, and this one maybe is splitting in 3. So we're going to continue to be surprised by that. So I don't want you to think that respiration, which is the thing that we talked about, stage 1 through stage 5, and photosynthesis, which is the second thing we talked about in plants and in bacteria that use light to make ATP. That's not the only game in town. There are a lot of other things that are going on. And other primary producers are things that make their own food. And other than plants, there are things that are making their own food.